Welcome back to Inside Biotech. I'm your co-host, Karishma Chugani. For those who may be new, Inside Biotech is brought to you by BCLA, a nonprofit organization dedicated to inspiring, educating, and connecting emerging scientists and entrepreneurs to help grow and diversify the biotech ecosystem in Los Angeles. On this podcast, we invite you inside one of the fastest growing sectors in Southern California. Here, we bridge science with storytelling to bring you the most novel, up-to-date, and thrilling news on the biotech world in SoCal. If you want to stay informed about current progress and developments in the biotech industry, you're in the right place. Twice a month, my co-host Riley Elmer and I sit down with scientists, founders, investors, and industry leaders to explore cutting-edge work happening inside their companies and to share diverse perspectives from across niches and career stages in biotech. In today's episode, I'm joined by Philip Sell, who received his Master of Science in Molecular Microbiology and Immunology from the University of Southern California and is now the CEO and co-founder of Metaba. At Metaba, Phil is working to bring innovation to treatment strategies for tuberculosis and other infectious diseases, while also leveraging metabolomics to help advance drug discovery across a range of indications. We dive into Phil's journey from an early interest in pursuing an MD PhD to ultimately choosing a different path, one that was focused on building and translating science into a real-world impact. He shares how his background in infectious disease research shaped his perspective and how he applies those skills in leading a company tackling complex, real-world challenges in antimicrobial resistance. We also explore the founding story behind Metaba, the scientific approach behind their work in drug-resistant tuberculosis, and the broader challenges of developing new therapies for infectious diseases. Finally, Phil offers thoughtful advice for scientists considering a transition to startups, having a work-life balance, along with insights into what it really takes to build and lead a biotech company. Thank you so much for being here today, Phil. Happy to have you here on Inside Biotech.
Thank you so much, Karishma. I'm happy to be here and honored to take part in the podcast.
Yeah, looking forward to having a great conversation. So let's dive right in. I know you originally planned to pursue an MD PhD, but ultimately pivoted. So can you walk us through your academic journey and what led you to make that shift?
Yeah, absolutely. The story, I think, begins, at least in my mind, the plan to go to med school since I was a very young child. My father was a physician and there's a lot of medical background in my family. So I had that influence in my childhood very early on. And so literally, since I can remember anything about school, I was like, oh yeah, I'm going to be a doctor. And so this was also an identity I had built up and strengthened over my whole journey through academics, through life, really. I always had a scientific perspective. And of course, it's always a non-traditional path, I feel like, where you're exploring and refining and then, maybe even redefining what you see for yourself. And so I guess more pertinent to the MD PhD, right? I had done that master's at USC, and that was primarily to bolster my MD PhD application and, also continue my education. I started to see things in how I can make an impact, right? Because that was the underlying, I think, drive for the MD PhD track in the first place, was that ultimately I wanted to have the largest impact with the research that I was doing. And so I thought naturally MD PhD, right? Translating technologies from the lab into the clinic, helping patient impact. Boom. And it's kind of a series of serendipitous events that kind of delayed my applications and showed me things in my career path that, you know, different signs that were pointing me in another direction. And that still wasn't an easy decision to make. But that's, I'll talk about meditation a lot too. I think having some sort of contemplative practice helped me recognize those signs and help me let go of this identity I had built up. But ultimately, I saw that I personally could have more impact by starting a company and doing my own thing than continuing on the PhD path. Because if I had chosen that path, I'd still be, another at least 4 or 5 years in school. So, so far, so good. I'm glad I followed the decision I made so far.
Yeah. Okay. So I know you mentioned you had a string of serendipitous moments. Was there a specific moment when you realized you were more drawn to building and translating science rather than practicing medicine?
Yeah, so I guess adding more to the details there on that. So, as I said, I started this master's program to bolster my MD PhD application. Most of that was because my undergrad performance wasn't maybe the most competitive to go directly to med school, and that's a longer story there. But focusing more on the master's and the serendipitous events there was first, I started in a virology lab. I love infectious diseases. I think they're super interesting, especially on the molecular side and the strategy. So I was going to join one of these labs and I leaned towards viruses. I thought they were more interesting than, some of the bacterial infections, which funny enough, right, I start in this virology lab and the principal investigator of that lab left after my first semester. And so I had to find a new lab, right? Because all the PhD students could transfer, all the postdoc, everyone could go with him, unless you were a master's student. They didn't have a master's program to follow. However, I wouldn't have continued on to Cleveland. I think I wanted to stay in LA and continue my master's here. I skipped New York in the first place to come here, so I don't want to go back into that climate necessarily. So did that my first semester and then had to find a new lab. And this one lab came highly recommended that was doing research on tuberculosis, which I was not particularly interested in. I didn't really know how big of a worldwide problem it was. Like, honestly, at the time, I thought, tuberculosis, why would I want to study this? Is this still even a thing? But the reason they came highly recommended was because they could do this technique of metabolomics. And I thought that would be a useful skill to learn. And not only that, going back to that original intention for the masters, a lot of scientists wanted to include metabolomics into their papers, into their research. So there's a lot of collaborations. Right? And if there's a lot of collaborations, then there's a lot of potential for publications. And if there's publications, that of course would bolster the MD PhD application, right? So thinking backwards, originally why I was interested in that lab. And then lo and behold, TB is super interesting, uh, especially from the metabolomics perspective. And this is kind of where Metaba essentially started. It wasn't initially in that lab, but in that lab is where I met my co-founder, the postdoc that trained me and taught me basically everything I know about tuberculosis and metabolomics. So we were very close, of course, having that mentor-mentee relationship. And then the next serendipitous event was, this was in 2021 after I defended my thesis. Instead of continuing on to apply for MD PhD programs, this opportunity came up to do research on coronavirus, right? And again, at the time, it's like the heat of the pandemic, and there's, a huge need for personnel doing this type of research. And I was basically already qualified in a way because working with stuff like tuberculosis and coronavirus or SARS-CoV-2 more precisely is, you work in this high containment lab, or in this case, the biosafety level 3. And even though Joey was still doing the tuberculosis research, and at this time I was doing the coronavirus research, we still had to remain in very close contact because no one can go in this lab alone. And so you always have to have this buddy system. And so we had to align our schedules all the time. And basically that's where like our morning conversations that was just mostly focused on aligning schedules always turned into, some of the visionary ideas that we had. And it just got to the point where we're like, hey, the science looks really good. Why don't we start a company? We both are kind of over academia at this point in our career and like give it a shot. Why not? So that's kind of the start of Metaba and also, pursuing MD PhD programs.
Yeah. No, that's amazing. I think it's all about the journey, and you don't know how your journey's gonna, you know, get you to your current destination. I always love hearing the stories that people have in their career and what serendipitous moments led to where they are now. And so, for listeners who may not be familiar with the company, can you explain what Metaba does and some of the core scientific approaches you're using?
Yeah. Yeah. I guess I can continue on with that story because that kind of explains our history, what we've done in the last like 3.5 years, and why we don't really have that much to show for it. But we started basically based off technology from that lab where I'd done my master's. And in that lab, we discovered a metabolic pathway that's basically a strategic shift in the bacteria that allows them to hibernate like bears, essentially, right? They feed off of the fat stores in their cell wall, which allows them to survive for long periods of time in very, high stress conditions. And so this is part of the reason that the current standard of care for a non-drug resistant strain of tuberculosis is about 6 months of 4 different antibiotics taken daily, essentially, give or take. And so that's a long time to be taking antibiotics. And so that's where this body of research came. And so we wanted to target that pathway and prevent this metabolic shift to happen to prevent that population from basically hibernating. And so that's where we started. And then the longer story short, in some of the metabolomics analysis services we had provided with what I call our sister company, Max Biopharma, who have been pioneering the field of oxysteroid therapeutics, doing some of these analyses for their programs, we were like, hey, why don't we test some of your compounds and models we're familiar with? And we ended up finding one that inhibits the bacterial growth. And over the last 2, 3 years, a lot of SAR studies, medicinal chemistry, improving on this compound until we have a pretty strong, potent and safe bactericidal compound, which of course is far more interesting to develop than an adjunctive therapeutic, which would have been that original target we were going after, because that metabolic pathway is only relevant during antibiotic treatments. Otherwise, it's not really relevant. So that's where we focused and pivoted into this directly acting bactericidal compound. And we've gotten it to first in animal studies, which, we'll have the data soon for the first validation, which is huge, very exciting. And so that's the therapeutic arm of what Metaba does. And, of course, it's our goal to address these huge problems, really humanitarian crisis worldwide. And that's back to that impact, right? It's like, I know it's maybe not the largest appetite in terms of market and, maybe the larger pharma players would be interested in. But for me, that's not what it's about. It's about the impact, right? Like, how many lives can I save? That's what we do with the therapeutic side. And then, focusing more on, I'd say, the diagnostic side, or really even just like the analysis services side. So as I said, Joey and I, we're both experts in metabolomics, and we have seen the need for this service in the market. So we kind of provide more of a white glove metabolomics analysis service. And a lot of the times, speaking to that need, like, we've never actually advertised our capabilities there, our services. It's really come to us from word of mouth. And, we provide it on a bandwidth availability basis as well. And it's another way that we can provide value to scientists, especially. But even branching off of that, there's forward looking diagnostics that I think can be built off of metabolomics broadly, generally measuring biomarkers, and then also, broader use cases in like the longevity, professional athletics, biohacker, health and wellness spaces. So that's something that we've been exploring more recently. And that's the short answer of all the things that we do.
No, that's very interesting. And I can also relate to some of your work in metabolic reprogramming because my PhD, some of the work I'm focused on is looking at metabolic reprogramming in Staphylococcus aureus bacteremia. So it is a very essential shift to be studying. What were some of the biggest challenges that you faced when you're transitioning from academia to a startup founder?
That's a good question. Challenges. I mean, I think I approach everything as like a challenge, right? There's no real roadblocks, right? Unless I decide to not approach it head on. And I don't feel like there's anything that I would say is challenging besides just founding a company in and of itself. Like, every part of that is not an easy thing to do, but at the same time, like, the challenge, like, that's what attracts me, right? If it wasn't challenging, I wouldn't be interested in doing it or trying to do it, right? And I think that was what interested me in science in the first place, right? Is that a problem solving aspect of like, okay, the challenge of answering this question, right? Like, how can we design this experiment to answer that and the proper controls to try to show causality as best we can and, like, contribute to the scientific knowledge. Like, I thought that was interesting and challenging. It's fun, right? Like, that's why we have these very long degrees and people dedicate their whole lives to research, right? And then I think when I got into the startup space, it kind of had a different energy than the academic research, not only industrial research, but also the startup entrepreneurial sort of community just has a whole different energy to it. And I think that was something that really attracted me. And then I guess also expanding that problem solving into, a ton of other facets, not just in the science, but also in the, regulatory, commercialization, like marketing, intellectual property, corporate structure, chips. Like, there's so many just different variables that you're juggling at once. And I really enjoy it. The challenge, I think, was thinking the different mindset you have as a scientist that is exploring like the fundamental knowledge versus a scientist that is trying to build a solution that is marketable, essentially.
I agree with that. I think as scientists, we're always attracted to solving problems. And if things aren't challenging, then we're not going to be very interested in it. So I can relate to that. So, in terms of, you mentioned that patients who have drug resistant tuberculosis can be on antibiotics for like 4 to 6 months, which is a very long time. Like on a biological level, or are you able to elaborate on what makes tuberculosis particularly difficult to treat compared to other bacterial infections?
Yeah, yeah, absolutely. Bacterial species, we have like two major types, right? Gram positive, gram negative. What that means is basically I don't know how technical I should be going into things or explaining it, but gram negative have this outer membrane essentially, and the gram positive have this sort of what is glycolipid, it's like just a thicker wall, right? And so those are typically the gram positive are typically harder to have drugs that are penetrating to places where they can act. Also, you have issues with drug uptake as well, and high enough concentrations for those transporters. So it gets very deep and broad in the level of strategic survival mechanisms. But fundamentally, tuberculosis is a mycobacterium tuberculosis. So it comes from not a gram positive, but it's almost its own version of that, which is even more difficult to penetrate, called a mycobacteria. And so the major thing in that mycobacteria have are mycolic acids, which are known to be like antigens, right, that can be recognized by the immune system. And so this is kind of like a twofold aspect that relates to this pathway. So when the bacteria are basically internalizing these mycolic acids to then utilize for energy and the creation of antioxidants, it's also removing these antigens from the surface of the bacteria. So not only is it harder for our immune system to recognize, right, because there's not as many flags to pick up on, but it's utilizing this. So it's trehalose monomycolate and dimycolate, which trehalose is like a 6-member carbon ring, and then it has this fatty acid chain, which fatty acid gets lopped off. It goes through the desaturase pathways that can also be generated for energy. But the main one we've been looking at was this trehalose, which is basically it'll go through trehalose synthase, which is this interconversion into maltose, which is the sugar that's able to be entering like glycolysis essentially, right? And carbon metabolism. And so the interesting thing about this also as a drug target aspect is that humans don't have this trehalose synthase enzyme, right? This is only an enzyme occurring in the bacteria, which gives a lot of benefits, right? When you're trying to develop a drug for that target. This is what happens when these bacteria, tuberculosis, Mycobacterium tuberculosis, are under environmental stress. So what is environmental stress? In most cases, in the pathological context, is nutrient starved and hypoxic conditions, which are found typically within granulomas. So nutrient starved, right? There's not a whole lot of nutrients. The major carbon source are cholesterol, sort of fatty acids, typically in the granuloma, and then hypoxic, right? Little oxygen. And so this is within the granuloma, which is a jail cell of immune cells. And so this is a normal immune response, right? Whether it's bacteria, virus, doesn't matter. The main ones are getting cleared out. Whatever is not cleared out is then fenced off in this little jail cell called a granuloma. And inside that, with these conditions, things will die off pretty quickly, right? But not Mycobacterium tuberculosis because it hibernates essentially. And so that's why this 6-month treatment regimen is required for it to eventually die in this granuloma. And so the interesting thing, I hope I explained the molecular aspect of it, but this whole body of research, like even the idea to look at this being what's happening was if we had patients that didn't fully adhere to that 6-month treatment, we'd see relapse in patients with bacteria that were genetically identical to the ones getting killed. And they weren't, in contact with other infected individuals. So the only like it was coming from them, right? And so typically we can point to a specific genetic mutation that confers a drug resistance. But in this case, they were genetically identical. So we're like, hey, what's going on here? And that's where diving into a deeper layer of the data, the metabolomics, this is where we were able to find and understand this mechanism. And so you, I'm sure, can agree with, the metabolic reprogramming and understanding like this very deep, nuanced data and how it correlates to the other data layers that you're probably looking at and how this can be informative, right, to make a drug at the end of the day. And so that's part of the reason, I guess the last aspect of that is like, okay, well, how can we make this type of data more accessible or more usable in a broader market? And that's kind of speaking to the recent pivot. So I know I moved away from the tuberculosis, but I get a little
No,
carried away.
that was great. So if I understand correctly, are you guys essentially going after like intracellular killing in a way? Because you said that these bacterium like hibernate in the granulomas.
Yeah, that's a good question. So, and then there it becomes very complicated because the primary cell type that MTB or Mycobacterium tuberculosis infects is the macrophage, alveolar macrophage. So not only are they in the macrophage cell, but they're also in the lysozyme, and there's different strategies around there. So there's like when you think about it intracellularly, there's multiple levels of penetration you have to have. Then you get to the bacteria with the mycolic acid and the levels of penetration there. And so typically that's a major cell type that they infect. But then you can also have essentially free bacteria in necrotic lesions. And I think these are the ones that are typically, captured by the granuloma. And yeah, don't quote me on that. But there's to say there's a very heterogeneous population during an infection. And basically when we have a antibiotic treatment, we're killing all of those ones that are not basically fenced off in the granuloma at that point. You can think of it like that, and that's why we've had a hard time with these tuberculosis patients relapsing or like these long treatment lengths because when we have that little bacteria in the granuloma, it's not actually detectable by our current diagnostics. So, as far as the clinician standpoint, they look like they're not infected anymore, and we kind of had to learn the hard way. The other aspect of that is I mean, I guess that adds insult to injury is the infective dose of tuberculosis is one bacilli. One single bacteria can produce a productive infection, where with a lot of other infections, especially viruses, it's like you probably need millions of particles to produce a productive infection. Infective dose is one single bacilli for tuberculosis, where, for a lot of infectious diseases, it can be millions of viral particles or what have you, to be able to produce a productive infection. So that's another important consideration with the difficulty of dealing with tuberculosis,
So
essentially.
it has a high bacterial load, if that's
Yeah.
correct.
Well, the small infective dose,
Yeah.
and it could have high bacterial load when you coughing up respiratorily, especially in the sputum, which is typically what they measure to confirm if there are bacteria or not, right? Which you wouldn't have probably a detectable number
Yeah.
if they're just within the granuloma and you're killing all the actively replicating ones. Yeah.
Very cool. Thank you for explaining that. Pretty fascinating to learn about the biology. So, do you see Metaba expanding its platform to address other antimicrobial resistant pathogens in the future, or have you guys already started doing that?
We hope to. We have in a way. So we have been testing other, like I said, gram positive, gram negative bacteria, and we haven't found anything, so we've found some things, but not strong enough signals that we can justify to pursue. And this kind of gets back to the difference of like academic science and industrial science, where it's like, okay, we can see some interesting things here and interesting things there, but then you really have to consider the amount of resources it takes to then further develop that idea. So that being said, we have tested the compound that we've been improving on in all these bacteria, and we've screened a bunch of other compounds. And that's one of the other interesting things with our lead candidate is that, not only does it not affect gram negative or gram positive, but it also doesn't affect closely related non-tuberculosis mycobacterium species. So this is the other very, very closely related mycobacteria that a lot of times you'll see similar drugs affecting this whole family, but our compound does not affect any of the other ones, which is actually a really great thing when you think about developing an antibiotic because you're not going to be also killing all the commensal and the beneficial bacteria, right? Which is why we see a lot of the side effects in these antibiotics. And that's why probably not feeling so good even after a couple days of taking antibiotics. So we hope to expand the platform. Of course, we need to have that first validation, and we hope to see that with this in vivo study that might give some proof to then, justify developing other things. But it's definitely something we hope to expand into for sure.
Cool. So we're going to pivot a little bit now into, startup and your biotech industry perspective. For scientists that are curious about startups but unsure where to begin, do you recommend any first steps they should take?
The first step that I think gave me a lot of value was just going out and networking and learning about like what people are doing. That taught me a lot. First of all, being a scientist and then getting into the startup space, I think it was extremely valuable that I could learn from others that were professionals in these specific areas, right? Like back to those other areas of problem solving, right? So there's a knowledge base, but then also that experience base. A lot of the AI tools out there, you can quickly, research markets, you can quickly iterate on products. So I think I would just get out there a little bit, talk to other entrepreneurs, like ask them about their experience because it's not easy, right? It's definitely not for the faint of heart if you truly want to go after this. And a lot of the times, like you'll almost know immediately, right? Whether you're willing to go at any length to try to make this across the finish line. I don't think most people want to do that, right? And there's always conversation about like work-life balance and these sort of things. And it's like, if you're going to be an entrepreneur, there's not a work-life balance, right? So you also have to consider your current circumstances in life. Like, you have dependents, you have people you have to care for. And that's like, there's so many important considerations before you make that jump. But I would say, yeah, just learn about the space a little bit. Ask people about their experience before making a big leap.
Yeah. No, definitely. What skills did you have to learn quickly as a founder that you say weren't part of your scientific training? Because it was quite a quick transition for you.
Yeah, it was. I mean, it's a good question. I mean, there's so many things I could point to. I think something that I learned quickly was like how to read contracts because that was like, okay, I have friends in the legal space and they're willing to share some experience there. I'm super grateful for the time and the energy they gave to me. Like I had one friend and he's like, hey, listen, like, let's get on a call. I'll give you like an hour or two. And what I'm going to do is I'm going to sit down and explain to you how to read a patent. Right. And we're going to go through this example and you're going to have pretty much like that base that you need to probably answer a lot of the questions you have. And if you need any questions past that, then I'm going to have to charge you. I'm like, all right, great. And so it's also good to, again, back to the networking, like meet people with those, professional skills that can share their deep insights into that area too. So I think contracts was a huge thing because I'd be bankrupt by now if I were paying lawyers to do all that kind of work.
Hello, I'm Dylan Kassane, host of Beyond Biotech, the weekly podcast from Biotech. Each Friday, we explore the latest in biotechnology, sharing insights on breakthroughs, research, and the innovations shaping the future of medicine. From advancements in gene therapies to AI-driven drug development and cutting-edge cell therapy, from top pharma and venture capital perspectives to fresh thought leadership from rising stars, we deliver deep conversations weekly with experts, entrepreneurs, and changemakers. Discover how biotech is changing the world, one innovation at a time. Subscribe to Beyond Biotech on Spotify, Apple Podcasts, or your favorite platform.
So, as someone, once again, who isn't in this startup life, what does your day to day life look like as a biotech founder?
I don't even know how to answer that because one day it's so different from the next. It's especially hard to answer now because we're kind of in this transitional phase, right? The last 2-3 years focusing on therapeutic, metabolomics here and there, and now really pivoting into trying to build a product around this metabolomics thing. So you can imagine the day to day in that 3.5 year span looks very different. And even if there's a similar theme, it's still different from day to day, right? Like one day I'm in the lab with the pipette in my hand, running the screens myself, right? Like I have to do this work. I'm not paying anybody else to do that, And then the next day it's like back to back meetings all day, right? I'm meeting with potential CROs or, maybe the next day I'm literally just like all day just writing a grant, trying to make a deadline. So it can differ a lot. There's not an easy way to to package that in. What does a day to day look like?
Yeah. Now, they always say when you're in startups, you always have to wear multiple hats. And I think part of your graduate training does help you in doing that, just because you're always managing a lot of different things at once. You have to learn how to problem solve and learn all your critical thinking skills and manage your time and try to have a work-life balance if possible. So, looking into the future, if Metaba succeeds in its mission, which I hope you guys do, I'm a big believer in your mission as well. But what impact do you see this having on global health?
Yeah, well, I mean, first of all, we could be saving millions of lives a year. with the tuberculosis therapeutic, and that can expand. You add a different antibiotic, and we're clearing out this human disease. I mean, not only we're saving millions of lives, but we're improving the quality of life. We're improving the time that families can spend with each other. I think that's an important thing to note. And I think it's actually a very good trend that I've been seeing more recently in the pharma space where, right, it's like, okay, let's treat the symptom, like let's pump this out, whatever. But they've definitely shifted to the patient experience a lot more. And so that seems to be the trend. I hope it catches and it really becomes like the centerfold because, right, that's why we're doing it. I mean, I'll speak for myself, right? Like some might be doing it like as a business decision sometimes, right? Unfortunately, as it is. But at the end of the day, it's like we want to help patients. We want to cure disease, or that's what I want to do. And that's why I want to address the underlying mechanism and actually have a cure, not something that mitigates the symptom, right? So I plan to approach therapeutic development with that single pointedness of mind in everything that I develop. So I know that the impact will be massive and I'll hopefully be checking off diseases as I go. I mean, it does take a while to develop a drug to market, right? It's probably over a decade from start to finish, if not more. So it's not easy to do. And then I also hope to see like the diagnostics change a lot too. And that kind of goes hand in hand, right? Because historically, I think we see a lot of these big blockbuster drugs that have historically been developed to address a symptom. And as our tools are becoming more precise, we can look more deeply into biological systems like metabolomics, for example, we can begin to see that, oh, this one symptom that we thought had this one cause actually could from 10 or 20 different underlying mechanisms, right? And that's why we see in clinical trials, like, okay, we developed something addressing this symptom, but then there's a percentage of the population that might not actually work. And it's because it's actually a different underlying mechanism that has the same symptom. So that's kind of how I see things going in the future. It's really going to be diagnostics that become the focus, right? As we can differentiate these mechanisms and then properly address what is causing that symptom, not the symptom, but what is it causing that symptom, the underlying cause, developing a cure. And that's where I think the pharma space will be a little bit more segmented for that reason as well. Yeah, I'm really excited to see where it goes too. We're living in really exciting times, the way that technology and data is just like blooming, like it's hard to keep up even day to day, like with the new things that are being released. So it's exciting times.
Yeah. Yeah, that kind of leads me to my next question. Like, where do you see the biggest opportunities in infectious disease drug discovery over the next decade? And what are your thoughts on utilizing AI and machine learning to help find this cure?
I see it a lot of ways. I mean, I hope infectious diseases will be a bigger thing in the next decade. I remember reading something, I think it said by the year 20, I can't remember if it was 2030, it was probably 2050, the number of deaths from drug resistant infections is going to surpass the number of deaths from cancer, right? Probably 2050 is more realistic. Infectious diseases has not really been a huge ticket. And you can see, a lot of people were focusing on oncology and CAR T, and now they're moving to like immunology. And there's like these big swings to what is big, GLP-1s, and now what's the next thing, and that's all well and good. But so needless to say, I'll go to these conferences and people will kind of be like confused, like, oh, you're working on not only just infectious diseases and not developing a vaccine, but you're like working on tuberculosis. Like, what are you doing? So, in the back of my mind, I'm like, just wait a decade. So, I don't know if it will happen in a decade, but these trends will eventually catch up to each other, right? If everyone's trying to solve oncology and cancer, right? Like, that problem is going to diminish. And if no one is trying to solve like drug resistant infections or not like a lot of money is going to solve this, like it's only going to get worse, right? Because it is. So we'll see. I think infectious diseases are super interesting. I think we could definitely do a better job of having a higher line of funding for preventative measures and forecasting and, these technologies because there's not a whole lot of labs that even research viruses. And I mean, that wasn't the case in 2021 around that time when I was working on coronavirus because like every lab I even knew of was like doing something coronavirus related, right? Funding spiked and it's gone and everyone forgets. And it's like, if we had that layer up a little bit more, we probably would have not lost so much in terms of lives and money and economics and everything from shutting down if we just had like a little bit higher baseline and people like continuing the research on that. So that's in the infectious disease area. And to the AI, I mean, that could be a whole another episode just talking about that. Actually, I posted something recently, but a friend, posted something about this model of a cell, and it's a very simple cell, but it was a very like seemingly comprehensive model where they're, accounting for a lot of different things. And it talks about like this model of a cell cycle of this very, very simple cell, like not a human cell, not a bacterial cell, but like the simplest of simple cell, which was a bacterium actually, but it was like a simple bacterium, right? And so modeling the complexity of this very, very simple cell over, I think it was like 100, I can't remember if it was 100 seconds or 100 minutes. I think it was 100 minutes. Modeling that took over 6 days of like constant compute at whatever level, and like the amount of energy that's being used and the cost to do that, to just model this little thing, and that is to say the modeling is improving, but we're definitely not there yet. And I can't predict like how fast it'll happen because things are moving very quickly. But as I said in the post, my takeaway from that post was like, okay, we've made really awesome advancements, but then also there's like these huge limiting factors in just the amount of compute that we have to model these things properly. And so the way that I like to portray that is literally just modeling a drug compound and a target, right, can be very computationally heavy because you're modeling 3D space, you're sampling 3D space rather. You're applying physics guided rules to this space, then you're also accounting for all the different chemical modifications, understanding the rules of chemistry there, and then how it's interacting with all these points. It's like it gets complex very quickly, right? And in ones and zeros, it's costly. And then get that to a cell where you have all these interactions happening all the time. And we're not even talking about an animal yet, because then you have to like, ingest it, or however you're taking the drug, where it's getting to, how it's metabolized, is it getting to the target tissue, then is it getting into the cell type that it needs to, affect, how is it affecting the things around there, is it the right concentration, before we even get into the cell where it can actually have that drug target interaction. There's so many levels of it. And again, it depends on what you're modeling. What you're modeling will depend on the question you're asking. And so there seem to be these initiatives to build, whether it's a digital twin or this like full computational model of just a cell, right? Of a human cell. And this is like being undertaken by many labs, tons of money are being poured into this. And I think it's going to be huge, right? I do suspect that we will have like a very functional model of even a single cell, also digital twin, looking back at like the Human Genome Project, right? It's like it cost so much money, so much time, so many people were working on just sequencing the human genome just once, right? Ever. And now it's like I could probably get my whole genome sequence for like $100 and I could get it like next week, Like it's insane how the technology advances. And so I think that's probably my guess as to what we'll see is like, okay, it's going to be really costly up front, but there's no shortage of capital going into training these models, especially in the LLM space. And that's a whole different story. But I think we'll see this start to become a really functional tool for biology. And, you can imagine how democratized that becomes in terms of models that we can use to develop new drugs and develop new solutions, whether it's a diagnostic as well. And I guess the last point I want to make on that, speaking of AI, also defining what we mean by that, right? Because, the general person saying AI is probably talking about a large language model, right? These are the most popular ones that people are interacting with, and understanding how a large language model might differ from deep learning, machine learning models, right? And then what is it that we're actually feeding the model? What is the desired output? And I think this kind of leads into where I see a lot of the future improvement happening. So I think, like there's clearly a lot of money being invested in LLMs and this whole like AGI race, and that's, whatever. That is what it is. It's happening. And I think LLMs will get to a point where the return is just no longer worth the investment in terms of the level of improvement it'll happen. And I don't know if we're there yet. I don't know when it'll happen, but I think we'll reach that. And that's when we have to start focusing. I think there are people already focusing on this. People have been working on this, but this is where I think it becomes more like that translational aspect of the deep learning, machine learning models in a specific dimension of data or of disease that properly incorporates the insights of that data to then draw like a meaningful, practical kind of like action. And so I think this is something I'm really interested in, especially applying that metabolomics layer onto the transcriptomics, onto the genetics, and then onto other modalities. And like, how could, like, I mean, we're in the day and the age of like all this data, all this AI, all this compute, all this analysis. And I think it's just a matter of time where we build an effective model that we will start to see patterns, probably with AI, right? Recognizing it better than humans. Like, we'll see patterns that become useful, right, for improving our health. So it's a long answer of it.
No, I think you mentioned a lot of good points, especially like it's important that you define the question that you're asking. What is the input to your AI model? What is the output? And I think even in my own research, you're getting different results with different machine learning algorithms based on the question we're asking, but translating that into the clinic, that is, I think, the tough part and how fast that can be done realistically, changing up the regimens that are already going on in the clinic. That is going to be the toughest part, I think, in the next decade. I think we have all this data, we have multimodal data, like you mentioned, but translating that to the clinic, I think, in my opinion, is going to be a challenge.
It's because we haven't properly modeled it computationally, especially, right? And even the animal models, plenty of cases where once we get into humans, it's a bust, more often than not. So I agree with you 100%.
Yeah. So you've been working on long and challenging problems like antibiotic resistance. So what keeps you motivated when working on these problems and, work-life balance? I know you mentioned that you like to meditate. So yeah, if you want to elaborate on that.
Yeah. So, well, what keeps me motivated is, I think, the mission and the vision, right? Whatever the task is, whether it is objectively mundane and laborious, I think I stay encouraged by the end goal. There's also a part of me that just loves the grind, like I feel good when I get things done, and I try to not like bring that to a toxic level because I fully recognize there can be that. And what helps me with that balance is meditation. 100% like most useful thing in my life to keep me sane. And even though I do work a lot, I also am very intentional with my time. There is time where, I'm relaxing with family or I'm going out to dinner with friends and like I don't have to rush because I've allotted this time for just personal relationships. But also, I play soccer regularly. I've been doing a lot of traveling, so I've been taking advantage of you know, the fact that I don't have to be in the lab and I can do a lot of stuff from the laptop. So I'm just all over the place, still working a lot, don't get me wrong, but also just working wherever I want to because I can. And so, I mean, I've made sacrifices to be able to have that life too, right? Like I've done things to make sure my overhead is very low. I'm not like living a lavish life in LA where like a latte is going to be $10. Like I can't afford to do that every day if I want to extend my personal runway to achieve these goals that I have, right? So there are sacrifices that I made. It's not all like, oh yeah, like I can just do whatever I want and do, I was telling a friend this the other day, and basically, my goals don't care about my excuses. And so that's something I think also very important in the mindset, right? Like if I'm making excuses, and I don't want to get into the philosophical aspect because then I can go into the whole like teleology versus etiology, and it's interesting, right? But like if you ask yourself, what do you really want, and like what are you willing to sacrifice for that? And I don't let distractions come into my life very often, right? Like I don't have any social media. My social media is LinkedIn and I rarely post. Like I made that post and I can't remember the last time I posted because over the last however many years that I have posted, it was always an intern that was doing it. So I really don't spend any time on that because I'm hyper focused on the things that are important to me. I know that if I'm effective in my work, I don't have to work all day. And I can also, I mean, I shouldn't be working all day anyways. Like, I don't think anyone should be doing that. But if I pick and choose, like knowing myself, right? Learning about myself, which I think ties back into the meditation, right? Like, what times am I productive? What times are good for me to do this? Just listening to myself. Like, do I need to rest? Like, I don't need to work today if there's some emotional stuff going on and I just need to like take a break. So I think for me to be effective, I need to be healthy. And that doesn't mean working. Like, time spent working doesn't equal success. It's like working effectively and strategically equals having that success in my mind. So it's important to do that because then you stay motivated. If I wasn't playing soccer and I wasn't physically active or like climbing mountains every once in a while because I love that, I love nature, like I will go insane if I'm not out in nature at least like once a month minimum, right? Probably more. But that's how I stay effective. And that's gonna look different for everybody. So that's the best advice I can give.
No, I agree with that. I think it's hard because I think in academia, like you said, it's like hours work, you know, might think that hours worked equals success, but I think the longer that you are in your programs and you start to learn more about yourself and, do the time management, you realize that no, it's important that you take care of yourself, number one. Like you have to be physically active and exercise, make sure you're eating correctly. And there's so much to it. Make sure you're, also socializing, like having that true work life balance in order to really be effective when you are putting in the work. So I can agree to that. That's something that I have learned over my PhD as well.
Good lesson.
Yes. Well, those are the questions I had for you. So if there is anything else that you want to talk about, the floor is yours.
Literally, I can literally talk about anything. So, I mean, I know this is a biotech focused thing, so that's why I try to stay more biotech. I mean, I'm happy to talk about anything. You mentioned time management a lot, and I know like a lot of the times because science doesn't move. Unless you've done science, you don't know how slowly it can move. And certain experiments like will take a long time before you get results. So it's a good time management skill to understand like how long projects will take, what that readout will be, how can I stack other things that could potentially like support that, but then also stepping back from that kind of bigger picture project management aspect. This is something I tell students all the time. It's like, why are you doing that step when you're in the lab? Why? What is that step? And then why are you doing this step? And then why are you doing this step? Like, if you don't know why you're doing each step, then you might have some problems when you run into a result that's unexpected, right? Which is always not a bad thing. But troubleshooting, as you probably found in the lab, is like a huge part of, I think, the education, right? Of doing things. And I think that also translates to the startup world too. It's like literally you learn by failing, I think. So I think that's an important thing, an important skill to have and not get bogged down in things not turning out as expected. I think that's become something useful to me is like, even though I've learned a lot of different things in these different facets of starting a company, it doesn't mean I have to be a professional in that to be effective, right? It's like know enough to be dangerous. And so like go back to the IP thing with the lawyers and reading contracts. And this is just my personal view, but like running a company, if I'm going to be working with a lot of individuals in different areas of expertise, like I also want to know enough about that to then guide them properly or lead them properly. Which my personal thing is like I immediately will admit like this is not my area. I'm not going to speak like authoritatively here because I just don't know. But if I do know, it's like I'm going to dig into understanding what is the actual problem. Let's get to that problem and figure out the solution, right? And the reason I can be effective in that with a group or whoever I'm working with is because I know enough about that area.
I think that's a great point because when you're doing collaborations, you're going to have strengths, you're going to have weaknesses, but let's utilize the strengths and work together. Part of that is understanding what your collaborator does so you can properly work together to reach the goal.
Yeah, no, especially with the collaborator. Like if you are working with someone that is doing what you said, Staph aureus, and you're working with this other like CRO that's like, hey, we're gonna run this experiment, we're gonna do it this way with this strain, and then we're gonna add this, and you're gonna be like, wait a second, why would you add this to this strain when it's not right? And you can only approach that if you have enough knowledge base to even ask the question. It's unfortunate I see this often where it's like people are spending a lot of money on CROs, especially in the biotech space, to do these studies. And you kind of need to be involved more than you probably think, right? You can't just be like, hey, this is what I want. I'm going to pay you for it. Do it. Right. It's like, no, like that's something I've learned too, is like, you can't just take everything for face value, take it with a grain of salt. Yeah. I mean, there's so many things I can share. I think another thing that I like sharing is kind of like the psychological aspect of business. And that's something I haven't really taken too many classes, like bare minimum, I think in college on psychology, sociology sort of things. But I've gotten most of my information from a podcast that focuses on sociology, psychology, and diving into the studies and the implications of it. And I think that was super useful for me to understand like how people think, like why people think the way that they do, what might be shaping those beliefs, but then also understanding like incentives. And I think balancing that and being able to read that early or being able to ask the question, like we're all humans, we communicate. And sometimes like a miscommunication can be very costly or waste a lot of time or waste a lot of energy or you get super upset. And it's like a lot of the times, like I'm just making stuff up in my head, really, if you think about it, right? Because we all have different perceptions. A lot of the times the perceptions are similar enough because this is of reality. And then that reality that I sense is then colored by my thinking. And my thinking is directed by beliefs that I hold from experiences, things that I've, you know, learned from whoever that I hold to be true. And I think that was a very enlightening realization is like, people hold a lot of different beliefs. And so that's why I really like to try to approach like the more facts, like it circles to everywhere, whether I'm working with a CRO, whether working with, an employee or a collaborator or just a friend, even communicate and make sure you understand before you try to find a solution because you might be totally misinterpreting what the problem is because there's these different, beliefs.
Yeah,
Um,
so many different viewpoint perspectives. It's hard to understand what someone is fully trying to communicate. So it's important to have conversations for sure.
Yeah, so I think that's huge. That's one of the big lessons. And then networking, like I've learned so much from other people, and that's also back to the communication too. And I've gained so much. I've gained so much from just being open-minded and even curious questions, right? It's like there's nothing I'm necessarily approaching with like, okay, I need to do this thing or that thing. And it's funny because I often get asked like, oh, so what are your goals here? When I'm at like an event or a conference or whatever. I mean, I'm sure it's probably good to go in with certain goals, but that's a hard question for me to answer because there's so many ways I can find value at this event or at this conference, right? And I don't know what to expect. And I think that's something that has served me well is like kind of approaching it. I'm not saying like don't have any goals, don't have any like goalposts to aim toward. I guess what I'm saying is like don't sell yourself short or pigeonhole yourself based on assumptions. Because I think if I were to have a goal, it's probably based on assumptions. I think I've been served very well to have an open mind and kind of just be led to where I'm supposed to be, if that makes sense, versus like going where I think I need to be.
Yeah. I think that's also just having being open-minded and not always being super fixated on the beliefs that you have and being open to what other people have to say and what other perspectives are. That I think that eventually shapes your own too. I think we covered a good amount and honestly like very different topics and different aspects, which is going to be like very interesting to the listeners, I feel like.
Thank you for, asking all the questions. I hope something was useful to the listeners, and thanks again for inviting me. I had a great time.
Well, thank you so much. I definitely learned a lot, and it was really great to hear your perspective, especially someone who was in academia first and that transition in like industry and a startup, but just your views on everything that we covered today. I think we had a great conversation. So, thank you for your time.
Thank you.
Thank you so much for tuning in to this episode of Inside Biotech, hosted by Riley Conover Elmer and Karishma Chugani. Inside Biotech is produced by Biotech Connection Los Angeles. I would like to thank our production team, Stephanie Wu, Serena Gao, Brian Jimenez, Danielle Ars, Danielle Liu, and thank you to our sponsors, Magnify at CNSI, USC Bridge Institute, KGI, LA Biospace, Technova, and Amgen for their support. For information about upcoming BCLA events, visit bc-la.org or find us on social media at Inside Biotech. If you are interested in partnering with Inside Biotech as a potential partner or speaker, contact our team at team@bc-la.org. We invite you to follow us on your preferred podcast service and leave a 5-star review. See you next time.